and I'm a scientist here in the laboratory for membrane trafficking, where I'm studying the link between Alzheimer's disease pathology and the degradation of material of genetics. Why are we interested in that? Because normally in cells like neurons we do not divide, there needs to be a very clear balance between the production of new components and organelles on the one hand, and on the other hand the degradation of all these components. And this is generally going wrong in neurodegenerative diseases like Alzheimer's disease. And one of the risk factor proteins that are linked to Alzheimer's disease is one of the proteins that is involved in this degradative pathway, and it's called phospholipase D3, or PLD2 in short, which I'm studying now. The interesting part is that it's specifically localizing in neurons and in these degraded vesicles where it's degrading DNA. However, as most people know, Alzheimer's disease is linked to the aggregation of proteins. So we question how is it possible that a protein that is involved in the degradation of DNA would actually lead to a pathology who is linked to the accumulation of proteins. So we answered this question into the paper, finding that actually PLD3 is specifically degrading mitochondrial DNA, so that's the only organelle in mammal cells that also have DNA aside from the one in the nucleus. And because neurons have a high firing rate and they do a lot of energy consuming things, they also have to recycle all the time this mitochondria, which also then bring DNA to the lysosome. And given the fact that in case of Alzheimer's disease patients, there is a problem with PLD3, that's the main degrader of this mitochondrial DNA inside these degraded vesicles, we see that it starts massively to accumulate in these vesicles, meaning that it's not only the degradative pathway of DNA that is going wrong, but that is just congesting the whole system and also affecting these proteins, including the APP degradative pathway that is linked to the Alzheimer's disease pathology. And we found that also that in that case, the lysomes get so filled that they actually start to leak stuff into the rest of the cells, which is of course bad because normally it should be contained inside these vesicles. And lastly then, we saw that this leakage is of course very wrong, and if you block it, it's possible to actually stop a kind of vicious circle, leading from extra material going to these recycling vesicles.